Hello everyone, my name is Claudia Martinho and I'm a postdoc in the plant science department under the supervision of Professor David Balcom. Thank you so much for watching this video and I hope you enjoy the Festival of Plants. So today I wanted to ask whether plants can teach us how to decipher the human genome and talk to you about my work in the lab and how I think we can make a small contribution at least to this question. So first of all, I wanted to briefly mention Gregor Mendel, who was a monk and a botanist that was very passionate about plants. And his experiments in pea plants helped us understand all the fundamental bases of genetic inheritance. And he would carry out many experiments where he would cross parents that have different traits, the expression of different traits, for example, as the yellow fruit or the green fruit. And he started observing that when he crossed individuals with two different traits, sometimes the progeny was 100%, so expressing 100% of only one of the traits. And he managed to explain that by coming up with the idea of genes that now it's so familiar to most of us. But at that time, Mendel didn't know what they were made of, didn't know how they were physically constituted. So this was quite impressive that he managed to work out and infer all of these based on crosses between different plants. So what explained that all the progeny displayed only yellow color was that the yellow color was dominant and the green color was recessive. And recessive means that the trait will only be expressed if you have two copies of that gene. So you could draw what we call in biology a Punnett, or genetics a Punnett square, but basically it's just writing down all the possible combinations of the genes and the specific copies that the progeny will inherit. So what was happening in the example I was showing you is that the progeny of the yellow and green pea results in individuals that carry a yellow and green copy, but because the yellow is dominant, 100% of the population expresses the yellow color. So much later, actually in Cambridge, people found that genes were physically made of DNA, and that DNA was made of these building blocks, and there are 4 types of building blocks, so T, A, C, and G. And if you would read this sequence of the building blocks, how they are aligned, you could just write them down, like if it was a word or a big sentence. And if this was printed out and put in a very large book, that would be basically what a genome looks like. For example, the human genome has, if it was printed, it would have approximately 262,000 pages. That's a lot of very thick books. And now, since many genomes have been sequenced because we have the technology to do so, we need to make sense out of all the sequence. And to make sense out of the genome, we can ask different questions. And one of the most intriguing questions for biologists is if the genome is the same in each cell type, what makes cells different from each other? And can the answer to this question help us decipher the genome? So basically, how people went about answering this question was by finding the position of genes and finding out whether they were switched on or off. And basically, when they are on, people say they are expressed because they are producing then the function. Whereas when they are off, they are just not doing anything. And if you have many genes in your genome, you can imagine that you have endless combinations of genes that might be on and off. And essentially, that's what gives rise to different cell types. And this idea has opened up the field of gene expression, which is the field we study in our lab. Of course, as you may imagine, this is much more complicated than this, and there are multiple mechanisms that enable to switch genes on and off. We still don't know all of them, and we still don't fully understand all of them. So there's a lot of work to be done there. But in our lab, we are interested in what we call epigenetic modifications, which are very intriguing chemical modifications that are attached to the DNA without changing their sequence. And that's why they are so interesting, because you have the same sequence, but a different expression of a gene due to the epigenetic modification. And when you attach these molecules close to a gene, basically you cause the gene to be silenced or switched off. And that's what I'm studying in tomato plants. So I'm studying this beautiful example in which a particular gene that encodes for the green leaf color is active in the green tissue. But we found that in some specific individuals that display this lack of pigmentation, which we call chlorosis in plant physiology, have the gene switched off due to these epigenetic modifications. Intriguingly, this epigenetic modification not only is inheritable, so it can be transmitted to the next generation. And this is something that is quite remarkable about plants. They can inherit epigenetic modifications, but also this epigenetic modification is not inherited in a Mendelian way, whereas others are. And that's quite interesting. So whereas, as I was explaining you, in a typical Mendelian segregation, what you would have if you would cross individuals with yellow leaf color with green leaf color, given that the green leaf color is dominant, you would expect 100% of the progeny to be green. But in the case of this particular example, some individuals in F1 display the silencing. And we don't fully understand what's happening here, but the assumption is that there is some kind of communication between the copy that was inherited from mother and the copy that was inherited from the father. And this is true if you do the cross the other way around. And that's exactly the aim of my project. I'm trying to better understand what kind of communication is occurring at this level and how are these epigenetic modifications maintained across generations. And this is important and could potentially have implications in the study of evolution, because as you can see, this is a population where the yellow leaf color is apparent in all of the plants. And the progress we have been making is that we are finding particular tomato plants that can suppress these so-called chlorosis or yellow leaves. So we are starting to understand this a little bit better. And one of our hypotheses is that what is occurring during this non-Mendelian inheritance has to do with how the DNA is packed inside the cell. And we are testing whether in the active allele, in the tissue that is expressing chlorophyll and the green color and has the gene that I mentioned before active, the mother and father copy of the gene would be quite far apart, whereas in the silence tissue, we speculate that they might be physically close and facilitate the transmission of these epigenetic modifications. So this is quite a novel and exciting field to explore, and it brings the three-dimensional confirmation of DNA into the picture, which we now in biology start to have evidence that can influence gene expression. So just to recapitulate, the fundamental laws of genetics were first established using plants as model organisms. Plants can carry epigenetic modifications, so modifications that are independent of the DNA sequence. And these modifications have the potential to alter Mendelian segregation. And we think that this alteration could be due to how the DNA is packed inside cells and the nucleus in particular. But above all, I hope I could convince you that we can learn many, many important and fundamental principles of biology by studying plants. So not only they give us food and many other fundamental materials for our existence and survival, but also they provide fundamental knowledge about biology, and in this case, genetic in particular. So thank you very much for listening.